Hello, everyone, and a very warm welcome to our first episode of the series Live from the Lab, Stories of Animal Ancestors. Today, our episode is on past and present of fossils and spiders. My name is Siri, and I'm doing my PhD project at the Natural History Museum in Berlin. I'm going now to pass you over to Milena, who organized this event with me.
Hi, hello everyone. My name is Milena and I am doing my PhD at the University of Exeter. And both me and Siri and most of our other speakers in all the episodes are doing our research within EvoCell, which is a Marie Curie network that is gathered with the tagline Animal Evolution from a Cell Type Perspective.
Most of the scientists that are giving presentations in this series are working in labs with cells and on living animals. But for tonight, we will start off the series with Graham Budd, who's a paleontologist and a professor at Uppsala University in Sweden. Graham is working on fossils that are millions of years old, even 100 millions of years old. And he's an expert in the origin and evolution of arthropods. It's a group that includes spiders, beetles, crabs, bees, and many more. Graham talks will be followed by Brenda Irene Medina Jiménez. She's a PhD student within Graham's working group, and she's working with many different living animals, including spiders. With her talk, we will make a transition into the laboratory. Furthermore, we have a very special guest tonight, Jason Dunlop. We couldn't get around not asking him to join this session, as he's been the curator for spiders and millipedes for the Museum für Naturkunde for more than 20 years. He's an expert on fossil spiders. But now to the moment we've all been waiting for. Graham, you have the honor of beginning the talks. Thank you.
Thank you very much indeed. Let's see if we can get this going. Right, you should be able to see my screen. I hope you can all see it. So welcome to this very exciting project we're doing all over Europe. And I'm a bit of an odd one out here because I am a paleontologist, as they already said. So I don't really work in the lab, but with our laboratory is the history of the world. So all our experiments have already been done, really. We just have to look back in time to see if we can find the results and try to interpret them. So of course we know that animal diversity is astonishing. There are lots of incredible animals out there, and there are millions of them as well. And this is a random slide showing lots of different types of animal diversity. Some of them you'll recognize, such as crabs or tigers. Some of them maybe a bit less familiar, like this. This is a snail. Some of them are extinct. There are some fighting dinosaurs here, Tyrannosaurus versus Triceratops. Some of them you probably won't recognize unless you're an expert, such as this strange blob here. So there's an enormous hidden diversity, as well as the more familiar types of animals we're aware of. And all this diversity started off from a single organism, of course, at some point. And it diversified all the way through time and space on the planet. And this is an incredibly exciting topic to study and try to understand from all sorts of different angles. Now, one of the things we know about animals, and that's what this whole research project is about, really, is that they're all made out of cells. So, of course, there are lots of different types of organisms, and we've been hearing a lot about viruses recently, and you're probably also aware of algae, plants, and bacteria. Animals are special for two reasons. First of all, they eat their own food rather than making it themselves. Secondly, they're multicellular. They're made out of lots of different cells. And since the 1950s, we've known an awful lot about what cells are like, because that's when people started using electron microscopes to get incredibly high magnifications of what cells were made up of. And they look a bit like this inside. They're really complicated little ecosystems, all of their own, really remarkable things. And the other thing that's amazing about them is how many there are. There are about 37 trillion cells in an average human being. that's, if you line them all up from end to end, that's about 3.7 million kilometers for one human being, if you just string all the cells out. And that will give you about 5 trips to the moon and back, just from the cells of one human being. So it's incredible to think that during our own development, we grow up from a baby to an adult from a fertilized egg, that all these cells appear and they all diversify into all the different types that they have to be. It's really, really astonishing. Because we know, as we saw from the trailer, there are lots and lots of different types of cells. As you perhaps know we have muscles, we have nerves, we have blood cells, we have bone cells, and so on. We have lots and lots of different cells. And many of these cells are shared by different animals, not just us, but we also find, for example, nerves and muscles in other animals as well. And trying to understand how the animals relate to each other and how their cells relate to each other is a really big and very profound problem in evolutionary biology. So, of course, we have some ideas about how the animals are related to each other. And this is a very simplified tree of the animals with some very technical terms in. But we're here, for example, this is chordates, that's the mouse representing the vertebrates, including us. This is insects are here, flatworms are here, jellyfish are here. So the big categories of organisms, of animals, we know roughly how they relate to each other. So all this tree represents really is a pattern of diversification that took place through time. Now, when did this take place? This is a big question, and maybe this will come up in the questions. But if you think about the history of life as a whole, it's a bit hard to squash into a single diagram. So you often see it as a spiral or something like this here. And so the origin of the world is down at the bottom of the spiral. The Earth is about 4.6 billion years old. Really, really old. And as we go through time, it goes spiraling up and up and up and up and up until we get to the last spiral at about 540 million years ago. And it's around then that we start getting the first really impressive, obvious fossils in the fossil record. Now, we have got fossils before that as well. There are some seaweed, some algae, and there are some single-celled mysterious organisms that are perhaps as old as 3.5 billion years old. But it's not until you get to about 500 million years ago that we start finding easily recognizable organisms, animals in the fossil record. And this is a really interesting period of time that lots of people study. So in fact, the oldest things we think might be animals are about 570 million years old. And they're really, really mysterious. So this is a close-up of one of them. It's from Newfoundland. This is a picture from my colleague, Alex Liu. It's called Bradgatia, and it's about 570 million years old. And you can see it has this beautiful fractal, multiply branching patterned surface, probably because it was absorbing nutrients from seawater or some other reason it needed to maximize, have a really big surface area. So we think that these organisms, although they don't look like anything living today, are probably the oldest animals we can find in the rock record, but they're very hard to understand. It's really mysterious, but they're very beautiful, and they're all over the world around this period of time. So this leads on to the really famous origin of animal diversity in the fossil record called the Cambrian explosion. So those really old fossils I've just shown you are very enigmatic. But after about 540 million years ago in the rock record, we get many different types of animal appearing really very quickly, one after another. And we call this the Cambrian explosion. The Cambrian is the period of time that starts about 540 million years ago. It's named after Wales, where these rocks were first described in the 19th century. And in very short periods of time after the origin of the Cambrian, many types of animals start appearing. And we see signs of increased diversity all over the place. Now, some of these aren't just fossils, but they're also the signs of the activity of animals. And these are called trace fossils. So this is a beach somewhere, maybe in Wales. You're probably familiar with these worm casts, they're called. And you know that these are the sediment that's been like squeezed out of the sand and the mud by worms that are digging under the surface. So as they dig, they push up all the stuff they've dug up through and make these little heaps called worm casts. And when the tide is low, you can see them all over the beach like this. Now, sometimes, of course, these get preserved, these structures like this get preserved in the fossil record. So these aren't animals themselves, they're signs of the activity of animals. And that's one of the most striking things you see in the fossil record, not just fossils appearing, but you start seeing signs of activity. So this is a really famous one, and it's really the fossil whose appearance heralds or marks the beginning of the Cambrian about 540 million years ago. It's called Treptichnus, and you can see it's a system of burrows. Some animal has pushed its way repeatedly through the sediment, through the mud, and left these little burrows, which have been preserved over 500 million years later. And you can see some other worm cast type structures here as well, very similar in a way to the ones we were looking at here. So we can begin to see, even if we don't see the animals themselves, there's something going on. But of course, that's not the only signs we see in the fossil record. We also see real fossils appearing. And for example, early on in the Cambrian, we start getting a diversity of shells made out of a variety of minerals. So these are hard parts, which preserve quite easily in the fossil record, such as this beautifully curved shell and various tubes. Many of these are quite mysterious as well, but some of them can be related to modern day animals such as mollusks and annelids, maybe. And we also find preserved body fossils, and perhaps the most famous one out of the Cambrian are the trilobites. And this is a lovely trilobite called Redlichia. This one is from, I think, China, photographed by David Rubkin. I actually have I have one here, in fact, to show you, if I can lift it up. So this is a fantastic set of fossils. I don't know if you can see them. From Morocco, these ones are. They're about 500 million years old, so they're a bit younger than the first fossils we see in the Cambrian record. See, they're enormous, and there's another really big one on the back as well. So already we see, quite soon after the beginning of the Cambrian, we start seeing some really spectacular and beautiful fossils appearing, which you can go and collect. So animals get big really quite quickly. The other thing we see in the fossil record is not just fossils like trilobites, such as this one here, but in some places, especially in China and in Canada, we see a few sites of what's called exceptional preservation. And there we don't just get shells preserved, but we get this beautiful soft tissue preservation. This is a soft worm that almost always would vanish without any trace, but just very occasionally, under really exceptional conditions, these worms get preserved, and other soft-bodied animals. And this tells us an amazing amount about early animal evolution. And one of the things we learn about the Cambrian explosion and the origin of the animals is, just like today, the arthropods become very common and very diverse very, very quickly. So this is an arthropod from one of the jointed-legged animals from the Cambrian. You can see the little legs coming off here and an antenna coming off here and the body down here. And sometimes you get beautiful preservation. This is from China, showing all the little jointed legs. So there are many features we can learn about these animals in terms of their general organization and the general structures they have. And sometimes we can learn astonishing details. So I don't know if you can see this one. This is from the same arthropod I just showed you, from China. And this black thing here is the nervous system. And I think if you look very carefully, you should be able to see these little black traces coming off. These are the little nerves coming off the central nervous system spreading into the tissues of the organism. So the fossil record is full of surprises. It can show us the overall structure and diversity of animals. It can show us even some tissue, some organization of muscles, nervous systems, and so on. But what we really want to do is try to learn what they might have been made up of. We can't really see cells in the fossil record usually anyway. So in other words, by looking at the fossil record, we get some idea of how modern diversity started appearing very rapidly about 540 million years ago. And that gives us a sort of a framework to try and understand modern diversity. But to go further, we really need to start looking at modern animals and relating them back to the fossil record. So nearly everyone else apart from Jason will now talk about looking at modern animals and their cell types to try and understand the origin of animal diversity from the other end of the story, 540 million years later.
Thank you for this very interesting introduction to the series, Graham. And we'll give the microphone to Brenda now. So Brenda, would you continue?
Yeah, I would like to share my screen now. So, okay. Yeah, so picking up on what Graham explained, from here in the laboratory in Uppsala University too, here I'm in the laboratory that I work, I will explain more about animal evolution and development using the pan-arthropods as a case study. Because as Graham mentioned, arthropods have been present since the Cambrian explosion. So what are pan-arthropods? Pan-arthropods are animals characterized by faculty of shedding their skin, which is properly called cuticle, after developing a new one underneath it. They are divided into 3 groups. The first group is velvet worms, the water birds, and the arthropods per se. So as Graham mentioned, arthropods are characterized by their jointed limbs. So they have joints that allow their limbs to flex, which is something that's not the case for the velvet worms and the water birds. Their limbs are actually stumpy and short, and they cannot flex. Also, because of this common characteristic between these 2 groups, they are commonly known as lobopods. Another characteristic of the pan-arthropods is that their body is segmented, which means that their body is divided into units that possess a pair of appendages each, as we see here and here. So my research study here, my goals are to understand the development of the pan-arthropods from an egg to an adult and discover the common and divergent mechanisms during their development to shed light into why are this big diversity of body forms that are within this group. And it's important to recap a bit into going back to the cell and understand certain concepts. So in every cell, starting from the fertilized egg, we have a DNA molecule that we inherited from our parents. And parts of the DNA, which are called genes, are transcribed or copied into another molecule, an intermediate molecule called messenger RNA. It's like the copied version of the DNA. And this molecule is translated into amino acids that later are going to produce proteins. So the proteins are the functional unit of the genes that have been copied. So what it means is that if a protein is produced, that means that the gene is active. So this is a concept that is very important to remember, because in the developing embryo, we have the cells that are developing, some cells that are going to form, let's say, the nerves and the nervous system. In these cells, certain genes are active that are not active in the cells that are going to form the digestive system. Here is a cell A, cell B. However, there's also some genes that are active in both of these cells during their development. So to see which one of these genes are active and inactive during development can help us understand more about the evolution of these animals. And what do we use to actually see this? We use model organisms. And in evolutionary developmental biology, or shorter EVODIVO, the most common model organism is the fruit fly. I'm pretty sure some of you might be familiar with it. And most of our knowledge, or current knowledge on the development of arthropods, it's based on the studies done in the fruit fly. However, in our laboratory, we do not work with the fruit fly, but we work with other arthropods. So let me introduce you to them. The first one is the red flower beetle, which is a known crop pest, and also is very much used in EVODIVO, kind of like almost as much as the fruit fly. As Brian mentioned, we also have beetle fossils in the fossil record. This one is 99 million years old. It's preserved in amber. Many insects are preserved in amber. The second animal that I'm working with is the pill millipede. Perhaps some of you are familiar with millipedes, but this millipede has a very peculiar shape because it can fold into a little ball, and that's why it's its name. Here we can see where it's pointed out in the center of the figure, a female that it's covering like a male, and they are actually mating at this moment. So we also can find millipede fossils preserved in amber. This one, for example, is 99 million years old too. And well, the shape is different from the one that I'll show you, so you can see the difference that happened through time. The other organism that I'm working with is the common house spider. I think we all are familiar with this animal. These are one of the specimens that I have here in the lab. And as we can see in the video, the small one is the male. He's trying to mate with the female unsuccessfully. Hopefully he has more of a bigger chance later. So when it comes to spiders, there are also spider fossils. And this one in particular is it's 110 to 113 million years old, according to the information I got. But more on spider fossils we will get from Jason, so please stay tuned on that. And finally, one of the representatives from the lobopods that I work with is the blue velvet worm. So as we see, the shape of these animals is what it's very, very important for you to remember, because the shape of the modern blue velvet worms, in this one in particular, it still looks like it has been conserved from the lobopodian fossils that appeared during the Cambrian explosion. And here we see one that dates to 500 million years old. And the body shape hasn't changed much in all this time. So that's very remarkable. So wrapping up, the reason why I presented my animals in that order is because the beetle and the fruit fly are grouped into, are closely related because they are grouped into a group called pancrustacea. That also includes crustaceans such as the shrimp and the lobsters and other ones. And the other animal, the millipede, belongs to a group called Myriapoda, which also includes the centipedes. These two groups are considered sister groups, but are more closely related. Then, like a little bit farther related to them is the Chelicerata, which includes the spider and the scorpions, the harvestmen, and other related animals. And all these groups belong to the arthropods. So the most closely related group to them, as I mentioned before, here is the onychophorans, in which are the velvet worms. So there are similarities that we can see perhaps in their external appearance, but there are also many differences. But how can we actually understand them better? Through the development of their animals and through these genes that are active or inactive that I mentioned earlier. And so currently, just as an example, I am working with a specific gene that it's considered a possible trunk selector. And what is a trunk selector? Well, especially when I want to talk about trunk selection or selection itself, selector genes are in those genes that are in control of the development of a specific region of the body, in this case, the trunk. And therefore, it was first found in the fruit fly embryo, of course. So how was it found? Using a laboratory technique called in situ hybridization. And what is this technique about in synthesis? In the cell that actually this gene is active, which means the process that I explained earlier, the transcription, the process in which the messenger RNA happens, is going to be stained. And if the gene is active, that cell is going to be stained. So when they conducted the experiment, they found out that the region that was stained corresponded to the trunk, but it did not stain the head. So it looked like it had some sort of discriminator role that between the head region and the trunk region. So I wonder, is this also happening in the research animals that I selected? So I used this technique in the laboratory to find where is this gene active, if it's active. And so in the millipede embryo, I saw in the region that is here highlighted, it's the head region. It's not active, but it is in the trunk. In the case of the spider embryo also, the head region, it's not stained. So it's not active. The gene is not active here, but it is active in the trunk region. And in the case of the velvet worm, also we see that the head is not active, but there is activity of this gene in other parts of the body. So it seems like the function of this gene seems to be conserved in arthropods and the velvet worms. And this dates from the Cambrian explosion. So we see that genes have conserved their function, in this case, a function for a long time. But there are also other genes that have changed their function. And so this is just an example to see how we can visualize that. Then further studies that I'm working on, I'm using actually new molecular biology techniques that will allow me to discover new genes that are active during the development of my research animals that perhaps there have not been described in the fruit fly. So that's something challenging and interesting. And later on, in our following talks, I think my friends are also going to talk about these techniques that also they're using when they're studying this diversity of the cell types. So, well, thank you. Thank you very much for your attention. I hope it was okay. I will stop sharing my screen now.
Thank you, Brenda, for your very interesting insight into your research. As a last and very important expert of our Fossil on Spiders episode, I would like now to welcome Jason Dunlop. Jason, can you tell us a bit more about fossil spiders?
Well, as Graham mentioned, there are fossils of spiders, and they actually go back over 300 million years. The oldest spiders that we know really go back to the Carboniferous. And there's actually more than you might expect. Most of them come from amber. Amber tends to be a bit younger, so there's only for the last 100 million years or so. But that's what's very nice about the spider fossil record is you can really compare that to what the molecular biologists are doing, and they come up with a tree of how they think the spiders split from each other. We can compare that with the fossils, and we see that, for example, the more primitive spiders like tarantulas tend to appear first, and then the more advanced spiders later on in the Mesozoic, around the time the dinosaurs were alive. And some of the spiders that be running around your house today only actually appeared in the last 65 million years. So they're actually quite young groups there. And you can sort of use the fossil record to follow this evolution of the spiders in really quite an interesting way.
Okay, thank you. And I think we can now start and open up our question and discussion round. And I think we have some very interesting experts with us. Once we have Graham talking about animal diversity, and he's an expert on the origin and early evolution of arthropods. Then we had Brenda talking about recent animals that she's researching on. And if we would have a timeline now, so we have the study objects of Graham in the very early beginning of animal evolution. Then we have Jason's study objects a bit more placed in the middle, and then Brenda with the most recent animals that she's looking at. So Milena, maybe you want to start off with a question, and then if you anyone else has questions, please put them inside the Zoom chat or inside the YouTube chat, and yeah, we'll be monitoring them.
Yeah, so I think I would like to start by asking about the very earliest stages of evolution. So my question is for Graham, if you could tell us more about the Cambrian explosion and why it happened, what happened in nature that enabled all those fossils to appear, and was it really an explosion, or have opinions changed on that?
Yeah, so this is a really big research question because, in fact, if we look at the molecules that are contained in modern animals and we try to work out how rapidly they're changing, we can then sort of work our way back in time and work out when they all started off, when they started splitting off from each other. And this is called a molecular clock. And these molecular clock techniques have been used to suggest that the origin of the animals was perhaps 700 or 800 million years ago, so much older than the Cambrian explosion, the actual appearance of the fossils we find. And of course, this is a big controversy that's going on at the moment, because if the fossils are right, then it suggests that the animals emerged perhaps younger than 600 million years ago, let's say, and then the Cambrian explosion of fossils is real, it's the real pattern of diversification. But if the molecules are right, it suggests that everything happened much earlier than that. And all that happened in the Cambrian explosion was really the appearance of fossilization. So maybe, for example, one of the famous examples is perhaps oxygen levels rose, and this allowed the animals that were already present to get much bigger, and then they start building shells, and we could find them as fossils. So there's a really big controversy at the moment. So if you want to ask what caused the origin of the animals, you first have to know when it happened. Was it, in the Cambrian or just before the Cambrian, or was it perhaps hundreds of millions of years before it? So I think, for what it's worth, that the fossil, I would do because I work on the fossils, so I think the story the fossils are telling is broadly accurate, and that the animals did diversify really rapidly about 550, 540 million years ago, and the fossil record is telling a kind of a true story. And then why did that happen? I think that's a really difficult question to answer, because one of the problems is we don't have a very good time resolution. We're paleontologists. Jason was talking about things being X million years old. I talk about millions of years. If you think in terms of ecology or the lives of all the animals we see around us, a million years is an unimaginably long period of time. We can't imagine what would happen in a million years. So it's really hard to work out all the little ecological and environmental changes that would take place even within a million years. And for me, a million years is no time at all. It's a tiny fraction of the amount of time we're talking about. So it's really difficult to fit the scales together. So that's something we're trying to work at, of course, but I think it's really hard at the moment. But what we do see is a very robust pattern of very rapid diversification. And I think that's quite an important result we learned from the fossil record.
Thank you. Thank you, Graham.
So here we have a question from John. Are there any organisms that seem more complicated than you would expect for the period they date from?
Well, perhaps I can answer that as well. So I think the answer is kind of yes, in the sense that what's always been very surprising about the Cambrian explosion is how rapidly modern types of animals emerge. I mean, the trilobites are not really modern types of animals, but they're pretty big and complicated already. From these exceptionally preserved places I talked about in China and Canada, we have animals that look like crustaceans. We have animals that look like many types of modern worm. We even have early, what look like vertebrates, which are our own ancestors. So it is quite striking how rapidly this diversification of body types and therefore of cell types took place, if we accept that the Cambrian explosion was a very rapid event. If it all took place hundreds of millions of years before, and it was all just hidden somehow, then it's not so surprising. So, but I think it is rapid, and therefore it is a bit remarkable, I think, the sheer diversity that appears so quickly, and how, pretty modern looking animals appear pretty quickly.
So it's truly fascinating how long arthropods have been around, in particular as well spiders, and who most of us have in our houses. And why do you think some animals do preserve their shape in such a long time, and then other animals get extinct or go through a rapid evolution, body and like in the body shape or wise? Why do you think some animals preserve their body shape for such a long time? Maybe Jason or Graham can answer that.
Well, I can answer from the spider perspective. What you would see is that some spiders, spiders eat insects primarily. And so you see the big jumps in spider evolution and spider body shape changes is very much connected to evolution of the insects and also to the evolution of flowering plants. So to begin with, the spiders were mostly probably just living on the ground in a burrow. The very primitive spiders today, they let little trap wires out of their burrows. And these, eventually once insects started flying around, they could bring these trap wires up into the air, as it were, and then you have essentially the first webs. And then when the flowering plants appeared, you've got the pollinating insects, and so there are many more insects flying around, and spiders developed many more ways of catching the insects. So I think the answer to this is really the ecology of the animals, or the ecology of whatever they're feeding on. And so when there's a change in the ecology, that induces changes in evolution in body shape as the animals start to utilize other resources, whatever they're trying to catch to eat, for example.
Yes, absolutely. And I think, for example, in the Cambrian, we find there are some shelled organisms called brachiopods, which they're not exactly the same as the modern ones, but they're quite similar, and they clearly have the same ecology. So they just found a kind of an ecological niche. They adopted it very early on, and then they didn't do very much after that. So they just found their thing they like doing. No one else kicked them out, so they just carried on doing it. So they didn't change very much. But as Jason says, whenever there's a big ecological change, then you get a lot of adjustments in the organisms as well.
Someone wrote into the chat as a question mark, evolutionary ecology of parasites.
This is definitely a Jason question.
Well, I can only again speak more from, for example, among the arachnids, you have a number of mites and ticks, for example. I mean, we're doing actually some active research on ticks at the moment. So we're finding the oldest ticks, which come from the Cretaceous period, about 100 million years ago, from Burmese amber. And whenever you're looking at a parasite, you've always got this interaction between the host and the parasite. So sometimes you can see the evolution of, in the case of ticks, the reptiles and the mammals, and when the mammals first appear, then you start to see the first ticks that would have been feeding on mammals. And so it's sometimes quite interesting to compare the evolution of hosts to the evolution of the parasites that are living on them. But in the case of something like a tick, of course, you can't follow it back too far because life all begins in the water. And so you wouldn't have had ticks in the Cambrian because you have to have something that could breathe air. For example, a vertebrate has to come onto land, probably in the Devonian or the Carboniferous, and then you'd have the possibility for an external parasite to live on this animal. How the animals evolved parasitism is another interesting question. it's sometimes the case that certain animals, some of these little animals like mites or fleas, started living in the nests of the hosts, and then by chance they were being carried around with them. This is called foracy, and foracy is like stowaways, so it's animals that sit on other animals and get carried along with them. And eventually some of them might have kind of come up with the idea, well, if I'm being carried around, I might as well have a little drink of blood while I'm on the way. And so that's perhaps one reason how parasitism would have first evolved.
I can just show you this picture. So just this year, some people have been publishing these really ancient examples of parasitism, even in the Cambrian. So this is a shell-like animal, the brachipod. I was just talking about those. These ones that don't change very much, and they seem to have all these tubes stuck on them, which are feeding off the feeding currents. So these are probably little worms that are living in the tubes, living off the feeding currents that the brachiopods, the shelled organism, is making. And what's significant about this is that if you look at the size of the shells that have the tubes on compared to the ones that don't have any tubes on, they're much smaller. So in other words, it looks as if it's definitely, this isn't just like living in the same environment, but they're having, it seems, a negative effect on the host organism. So this is the first example of like really negative interactions of parasitism in the fossil record, based on the idea that the shelled organisms can't get enough food compared to the ones that don't have an infestation because the parasites are taking it off them. So we can see that, again, this is, these quite complicated interactions started off pretty early on, already 520 million years ago or so.
That's wild. I was going to ask you and Jason afterwards, what is the strangest fossil you've encountered, but I don't want to put you on the spot and make you think of something better than this. So I'll go back to the molecular sphere and ask Brenda. So you showed us how different animals have very similar genes regulating their development, and I want to ask why does that happen? Why is development so similar in different animals, and what can we conclude about evolution from that fact?
Well, the thing is that well, in synthesis, it works. So those genes actually are active in there, or like are part of these networks that are benefited also by the ecological circumstances of the animals. So they are kept like throughout time. So it's like the presence of the gene that I mentioned, it interacts with other genes, and sometimes it interacts in a different way in other steps of development, because I show, for example, from one step of development, but then later on it can change too. So that depends much on how it is going to produce the animal later on. And evolutionary biology actually help us exactly to analyze these relationships between the genes and how much they have changed between the groups. And when you compare them, actually you see that if this relationship have been kept, and the reason why is because it's actually beneficial for the animal. Like if that shape actually, like the shape of the animal actually, it's actually helps the animal to maintain it and during time. So it's going to be kept. So okay, let's keep expressing this gene. And if not, it's going to be blocked because later on it's going to acquire a different activity on a different structure, so that we see that the gene is not active in the same way, or like not expressed in the same way, and interacts with the other genes instead. So it is like that.
Okay, sorry, there's another question for you, Brenda.
How did you choose?
Yeah, more. How did you choose the specific animals? So the group or the species for your project, so more based on practical aspects or scientific arguments?
Well, the scientific arguments are that these belong to certain subgroups within the arthropods and the pan-arthropods. So like the globopods, like I mentioned, the onychophorans, So these are kind of representatives of all these groups, but also, as the question points, it's a practical aspect that I have the resources to actually breed the animals here, because since I'm working with their embryos, I need to have the conditions for them. And right now I have the conditions for this type of animals, like to breed them. Like these animals are terrestrial, so it's like, it's a different thing if I decided to work with marine animals. Like in the following talks, our friends are going to talk about marine animals and how they actually have to breed them, and it's a different thing, it's a different challenge. And in our laboratory, it's easier to work with terrestrial animals like the spider. It's something that is very easy to work with in that respect. When it comes to the onychophoran, like the velvet worm, it's a bit of a challenge because we have to bring them from New Zealand, but and that's a bit of a hassle, but it's a challenge too. But we had the chance to actually work with them too. So it depends actually. But the reason why I actually chose them was exactly that. We have the conditions here in the lab, and also they are representative within the phylogenetic tree. So that's why they're investigating us, are gonna give us answers that are to the questions that actually we're asking. Yep.
Thank you, Brenda. I have another question for the paleontologists among our speakers, and I would like to ask you, what do you think in a million years, when alien paleontologists look at the fossils from our era, from the Anthropocene, what will they think of this time, and what will they think of us?
Well, so the Anthropocene is this idea that humans have changed the world so much that a new geological period should be considered to start at some point in human history, although it's a bit argued about what that moment should be. Well, I think it depends how long humans. It depends how long humans are around for, really. But remember that the timescale is measured in millions of years, and human culture has been around a few tens of thousands of years. So we're going to make a very, very small line in the rock record so far. There'll be perhaps some bizarre uranium or radioactivity spikes in it, depending on how we wipe ourselves out. Some really strange geochemistry. And I think apart from that, it'll be really hard to piece together what happened in this, what might be a very short interval of time. Hopefully we'll go on much longer than that, and then they'll find all our other stuff as well, all the Coke cans and planes and all the stuff that will be just jumbled into the rock record. But the timescale is very condensed normally in the rock record, so everything will be there really squashed to a very small amount of space, and it'll be really hard to work out the history of this very short period of time so far.
Laura was commenting that she does wants to know what the oldest fossils are that you found so far. Can you comment on that?
Yeah, okay, I can show you, perhaps if I can show you on my mobile. This is a picture of a spider from Burmese amber called Chimarachne. If you look very carefully, you can see it's actually got a tail. And this is actually essentially the most primitive spider that we know about. And what it tells us is that the very first spiders that we ever found, or that lived, would have had a long tail, like some other groups of animals called whip scorpions today. And this was a fossil just, or the pictures were sent to me about 2 years ago. And it was one of those moments where you literally, your jaw drops and you fall off your chair with surprise, because this is such a remarkable missing link between spiders and, as it were, more primitive groups of arachnids. And then this is really a very, very fascinating fossil, because it's got some of the key features that you'd expect from a spider. For example, the spinneret, so it would have made silk. Whether it made webs, we don't know, but it certainly would have made silk. But it still has these very strange features, like a long tail at the back. And it's really, as it were, sort of like a kind of, for me as a paleontologist, I always wanted from the start to say, what will be the origin of spiders? And now we found it. We found an animal that looks, or at least has the body plan of what a spider ancestor would have looked like.
Sorry, well, how old is it, Jason?
It's 100 million years. It's Burmese amber. It's like a late survivor. We know that animals a bit like that were known from the Carboniferous, so they would have split off from the rest of the spiders much earlier. And I mean, this was a little bit like finding they say like a dinosaur in your garden. You were finding an animal that really shouldn't have been there, but it somehow survived right through to the Cretaceous. So it was running around under the feet of the dinosaurs, even though it's a much, much more primitive animal that should have evolved and disappeared 300 million years ago.
Actually, I remember like when looking for examples of the spider fossil, I found one that dates from apparently 430 something million years old, and it's quite controversial because of that, because it's too old for what like we usually have in the record. It was found, there's a formation in the Jinju, I think, in South Korea. It was found last year, and that was quite like, it made the news. I saw like several news. Like, what do you know about that? That's like, that what caught my attention a lot.
As far as I know, the oldest genuine fossil is about of a spider is still only about 312 million years, something like that. The oldest arachnids go back to the Silurian. So you've got genuine arachnids that are related to spiders going back to about 410, maybe 415 million years. But anything older than that would be much too controversial. There was talk about a mite from the Ordovician, but I don't think this was shown to be a contaminant. It wasn't a genuine fossil.
I saw the publication. It had like a register of several of these spiders that they found. Interesting.
Okay, just quickly, we've reached the end of our hour, but there are a couple more questions. So I suggest for those who do need to leave, you can leave now, but I welcome you all to stay just a little bit more longer so we can answer all the questions. Let's say another 10 minutes, if you're okay with that. And I will just share quickly a link to a survey that you are welcome to fill out. We are very interested in your feedback and suggestions for the development of an online exhibition on the topic of this series. But yeah, let's continue with the questions. Darnell, I want to know about use of paleoproteomics to reveal the evolution and extinction of ancient parasites.
Yeah, I'd like to say something. I mean, so in general, studying parasites in the fossil record is quite difficult because parasites tend to be very soft-bodied inside other organisms, sometimes such as lots of these trematodes and other flatworms and stuff. So they can be quite hard to study, although there is quite a long list of fossil parasite interactions through time, including specific types of deformations you see in the hosts and so on. In terms of paleoproteomics, the trouble is that proteins and other biomolecules in general aren't very robust, it turns out, in the fossil record. So it's been quite hard to extract reliable information of that sort from the fossil record. But having said that, that study is kind of in its infancy. There are certain types of molecules such as porphyrins which have been potentially discovered. So there's a long way to go with this story. But in general, you know, the big biomolecules such as DNA seem to break down really fast. Proteins also, the best we can get are some remnants that might give some kind of fingerprint or sort of hint of what they used to be. And maybe in the end, you'd be able to say something about parasitism from that. So that's certainly something that's possible, but I don't think anyone has done it. So the best things you can do at the moment are like matching the phylogenies, so the trees of the parasites and the hosts, trying to put them together, and you might be able to then work out when it was that the parasites started inhabiting the hosts. But some of these parasite stories are really weird. Like there are some parasites in the Cambrian, the pentastomids. They look just like the modern day ones. And these are parasites of dogs and cats and snakes. They live in the throats of vertebrates. Occasionally even humans get them. I hope I never get one because they seem disgusting. And yet there are quite big ones in the Cambrian, and they can't have been in reptiles. So what were they doing then? It's really unclear. So there are some big mysteries we don't understand about parasites.